which, thanks, you're good. And our lab specializes in forest community ecology, a lot of forest dynamics and things like that. And my dissertation overall focuses on the ecological impacts of the pawpaw tree, Asimina triloba. A lot of you are probably familiar with the pawpaw tree because of their local edible fruits. But not a lot is researched about their ecology, so I was excited to dive into that. All right, so the inspiration for this project comes back when I was fresh out of undergrad in 2019, and I was actually working as a technician on Jonathan Meyer's forest geocensus team. So we were out there every day measuring every woody stem over a centimeter in diameter. And our team noticed that when we came across smallpaws, they always seemed to happen in these big patches, and there seemed to be no other tree species that grew within these patches. And on top of that, the understory seemed pretty bare as well. But these were still anecdotal observations. It hadn't been tested yet, and I just sort of became obsessed with trying to understand why pawpaws are the way they are. In case you're not familiar, here is a little bit about our focal species. So they cover basically the entire eastern half of the US, which is surprising given it's from a tropical family, Ananaceae. It's like almost entirely tropical plants. This is the northernmost member of that family. And while they have a broad geographic range across the US, at smaller scales, like this map in the middle here, they have really clumpy distributions because of those clonal patches they form. They can reproduce sexually through their flowers and their fruits, but they also reproduce clonally underground, and that's why we have that kind of patchy structure. And that's great for me because it sort of sets up a natural experiment where either pawpaws are present and they're present in big numbers, or they're just not there at all. So I decided to capitalize on this, and in the summer of 2021, I did a study where I surveyed 25 plots outside of pawpaw patches and 25 plots inside of pawpaw patches. And there were 1 meter plots. They're pictured here. Hopefully you can tell there's a bit of a difference between the two. The one on the left had way more species, and in addition to having more species, there were more individuals of those species. And in case you can for the graphs and not the pretty nature photo, I put some graphs up here. Basically, the one on the top shows that when we look at both woody species and herbaceous species in the understory, there's higher species diversity outside of pawpaw patches. So those are those yellow blocks versus inside the pawpaw patches. That green block is lower. And even when you look at just the herbaceous species, so forget all those tree seedlings, it's still same pattern. Part B of this graph is in regards to community size. So that's just the number of individual plants that are in these plots. Again, it's a similar pattern where there's a higher community size outside of the pawpaw patch versus inside. Great. So we established that a couple summers ago. So we're like, okay, why is this happening? Let's try to look at light and soil and some other things as well. I also wanted to look at whether or not the edge of the pawpaw patch was basically having edge effects. So where 2 different kinds of habitats meet, sometimes funky things happen ecologically. So even though it's all forest, maybe there's edge effects caused by these pawpaws. I had another great team of undergrads help me that summer, which was great coming out of the very lonely pandemic. Fieldwork is fun, but it's much less fun when you're doing it alone. So I was very grateful for that. And we were able to get funding to buy one of those lenses to take the fun canopy photos to analyze for light, but that's still in progress. The other part that's still in progress is the soil sampling in particular. And I wanted to mention this because we collected 140 samples this past summer and shipped them off to Missouri University for nutrient analysis and soil particle analysis. And I was able to get some funding for this, which is going to be such a huge part. So I was really excited that we had the funding to do that. So we don't have to look at each individual bar here, but the general trend is that on the bottom, we have the names of species of plants or their species codes. And on the y-axis, we have the frequency of those species. So just the number of individuals in the plots that are outside the pawpaw patch minus the number of those species inside the pawpaw patches. And you'll notice a general trend. Most species have a higher abundance when pawpaws are not in the picture. And of the species that are present within pawpaw patches, there's only one that was consistently more abundant inside pawpaw patches, and it was a fern. So if you're familiar with lowland brittle ferns, Hysopteris protrusa, that was the one species that we had that was consistently more abundant when pawpaws were more abundant. But we wanted to know, why are there fewer individuals of plants in pawpaw patches? Is it because plants just aren't dispersing into the patches for some reason? Is it because they're arriving there? They can disperse there just fine, but they're not germinating. Maybe they get through those first steps and they're just not surviving far enough into adulthood. Or maybe they make it into adulthood and they just never flower or go to fruit. So there's multiple kind of barriers to providing a sustainable population inside these pawpaw patches. And we really wanted to figure out, where is the barrier in this whole process? So we chose 3 of those species that were rarer or had lower abundances inside the pawpaw patches. And if you know your forest plants, you'll know that they're not very exciting plants. They are everywhere. So we have Persicaria virginiana, which is known as, sometimes it's called jump seed, sometimes it's called knotweed. The middle one is Hylodesmum glutinosum. It's a bean. I don't know if you go for hikes and you come back with all those triangle-shaped seeds on your pants that are a pain to get off. That's that plant. And the last one is Adriatina altissima, which is sometimes called white snake root. And it's an aster that blooms, it's starting to bloom right about now, all the way into late October. So we chose these 3 species to plant in some experimental plots. And our planting design looked like this. So each species would get 2 plots inside and outside. Oh, yeah, sure. Oh, yes. So these were species that we found in the previous 2 summers that we knew were more abundant outside the pawpaw patches. So that was criterion number 1. Criterion number 2 was that they were still present within the pawpaw patches, but in lower abundances. And 3 is that they had seeds available for purchase. So these were the only 3 species I was able to find for that. That's a great question, thanks. Yeah, so both inside and outside, we had 2 plots where we planted seeds for each species, and 2 plots where we were going to plant seedlings of each species, just in case something went wrong and the seeds didn't germinate. You know, we were going to grow the seeds that we ordered in a greenhouse, but there was a change of plans there. We'll talk about that. But overall, there would be 15 plots inside and outside, and then there would be 5 sites replicated at Tyson Research Center, which I understand some of you have visited before, but it's out there in Eureka. It's where I do all my research. It's my home away from home. And then of course we had some plots where we didn't do anything to it. Gotta have those controls. So our step 1 was planting the seeds. Like most things in science, this ended up taking a lot longer than I was anticipating, because we were first had to stratify them according to the seller's instructions. And then each plot got 200 seeds. And I wanted to make sure that the seeds that we ordered and went all through the effort of stratifying were actually reaching the ground and not just sitting on top of the leaf litter. So we had to go through and remove the leaf litter and plant the seeds in each individual 10 by 10 centimeter cell in this meter plot. And as a spoiler, only one species germinated. I would be interested to hear if you all have a guess of whether you think it was the aster, That is an excellent guess. And the data that I know Missouri Botanic Garden has put out would say that like white snake root definitely germinates. She does great. She goes hard, but it was actually the bean. Yeah, so we're still holding out hope that the other two species might germinate next spring, but you'll have to stay tuned for that. And I did want to thank the people that helped me plant these seeds when I panicked and realized I wasn't going to be able to do it by myself. So step 2, we said we wanted to have plots that had seedlings in them, not just the seeds that we planted. We ended up going around Tyson Research Center and digging up baby plants of these 3 target species, which was fun. These are my 3 undergrads from this summer that were just a massive help to me. So after we dug them up and put them in little containers, we set up a shade tent, a very makeshift shade house, to test whether or not they could survive that initial transplant shock. After a few weeks in there, we took the survivors and we distributed them to their respective plots back out in the woods. So there were 11 of the aster, Adrutina altissima, per plot, 7 of the bean, and 5 of the jumpweed or knotweed, for a total of 58 transplant plots. So it ended up being a lot of digging. I think about half of them have survived, several months later. So not bad. I ended up being pretty pleased with how this turned out. All right. So the next most important step is to finally monitor all these plants that we put in the ground one way or another. When we went to go visit our 150 plots, in each one we took a count of the emerging individuals. So we had a height cutoff based on their description in the Flora of Missouri for what their like maximum adult height was. So if they were emerging, so under that adult height, if they were an adult, or if they were flowering. And that was for all 3 target species. Then we recorded the percent cover. We went ahead and recorded the heights for the transplants just to see how they were doing. We visited roughly every 2 weeks, and we were able to complete 4 visits. So I was really excited about that. Visit 5 is still in progress. And once all of the visits are done this year, I'm going to count the seeds that the flowering plants produce. In order to do that, again, with Wigness grant money, I was able to buy these organza bags. They're like little party favor bags, but I was able to get really big ones to go over the flowering plants. So about a week after I noticed that an individual is flowering, I go back and put the bag on. So I make sure it has time for pollinators to visit and for the flowers to finish blooming. I wait essentially till it's just about starting to go to seed, and then I put the bag on so that predators or wind, whatever, can't carry off my seeds that I want to count. Okay, so this is just year 1 of data collection. I'm going to do even more data collection next summer. And I've been working hard to crunch the numbers this week. But again, like most things in science, this is taking longer than I anticipated. But I do have some data for you. So the beans, this is the Hyloidesmum glutinosum. O stands for patches, and P stands for pawpaw patches. That's where there are pawpaws. And the colors are the 5 different sites where I conducted this experiment. So you can see there seems to be, consistent with what we found earlier, there seems to be more individuals outside the pawpaw patches, but you'll notice there are no error bars, there are no statistics, so I can't say anything confidently yet, but there does seem to be a promising pattern there. I did want to shout out my undergrad students. This was a really rewarding part of this summer. They each had individual projects that we also worked on. So I had one student, Liana Van Zen, work on birds. They surveyed the birds inside and outside of pawpaw patches. They ended up finding that there wasn't a big difference in the composition of birds at that small of a scale, but they were also able to find surveys all the way back from, I think, like the 1980s. And they were able to compare those surveys and find that there seems to be a change in the composition of bird species at Tyson. So they're hoping to continue with that research. In the middle, we have Maya Irvine, who is looking at the understory composition of plants at the intersection of pawpaw patches and honeysuckle. So she's really interested in invasive bush honeysuckle, and found some really odd results about areas where there was neither pawpaw nor honeysuckle, places where there was just pawpaw, places there was just honeysuckle, and places where both pawpaw and honeysuckle resided. In terms of number of individuals, so that abundance measure, nothing was different from each other except those combined patches where there was both pawpaw and honeysuckle. So they're doing some sort of weird interaction to affect the understory, which we would love to continue looking at. And finally, Mary Ruse looked at, those are all pawpaw cookies over there. She looked at the correlation between the size of a pawpaw, its diameter at breast height, and its age. Our oldest pawpaw was, I believe, 38 years old, but it was only 6 centimeters in diameter, whereas our largest pawpaw was 11 centimeters in diameter, but it was significantly younger by about, I think, 4 or 5 years than the oldest one. So it was double the size of our oldest one, but younger. It was really bizarre. So basically we concluded we can't guess a pawpaw's age based on its diameter. And also, just as a shout out, the Tyson Research Center has an Olympics competition at the end of every summer, and my team did get 2nd place. So we were quite proud of that. Thank you. The conservation implications of this research, I'm really hoping that knowing how pawpaws affect these other species and at what life stage those species are affected will have meaningful conservation implications. So if pawpaws decrease dispersal, so if they're limiting plants from even arriving in those areas, then it might be important target areas for land managers to put seed addition treatments. Whereas if it doesn't matter how many seeds disperse to a pawpaw patch, they're not going to survive. Knowing that, we would then know, let's not waste our precious time and financial resources in adding seed additions to those areas. And depending on the, the management strategies or goals of that area, maybe you thin pawpaws out. I don't know. But yeah, hopefully it'll have some real world helpful impacts. And I did want to shout out the middle drawing is done by someone who used to work for the Myers Lab and is now my close friend and roommate. And she sometimes goes to art festivals and sells things like this. So those are all Missouri native tree flowers. All right. I want to say thank you again. The funding that I've received from WIGNES has been very helpful in soil samples. We've got toothpicks to help keep straight all our teeny tiny individual plants that are sprouting in these plots, and the organza bags to save the seeds that I'm going to be counting. So with that, if you have any questions, I'd love to hear them. Yeah.
Do any soil samples in the pawpaw?
Mm-hmm. Yes, definitely. Yeah, so we were able to get soil samples from 140 different plots, and those were split pretty evenly between inside pawpaw patches and outside pawpaw patches. And some of them we got right on the border there to see if right inside is different than right outside. So I sent them a couple of weeks ago, and I'm still waiting to hear back the results. Yeah, yeah. Mm-hmm. Mm-hmm. Yep, yeah. So my student that was interested in honeysuckle in particular was very interested in allelopathy. I've looked into it before, and the literature seemed to say, no, pawpaws are not allelopathic. We found one paper, my student Maya and I found one paper from the Ukraine saying that maybe they're a little bit allelopathic. The soil samples that we collected this past summer won't get to that question, but some research that's being done by my advisor and his collaborator do deal with the compounds that are in the leaves, and we might get a better idea from that project. The soil samples that we sent off are going to be looking at nutrient analyses. So things like phosphorus, stuff like that. We couldn't look at nitrogen because nitrogen can change so much even day to day that the methods just, you'd have to measure nitrogen basically in the field several times throughout a whole year. So that was kind of off the table for us. But thankfully, the literature shows that doing this soil particle analysis, so how much is sand, how much is silt, how much is loam, is a similar proxy in terms of telling apart really important, ecologically important differences in soil. So I'm excited to get those results back.
Yeah, why did you choose like
Yeah, so the spring flowering plants, largely because that first summer that I was collecting data, I didn't get going until about June or July, so I missed the spring flowering plants from that one. Secondly, because a lot of spring flowering plants are ephemerals, there's a much shorter window to monitor them, and that just logistically is more difficult. So that was the second reason. The third reason is because of the spring flowering plants that I was able to monitor and capture, I couldn't find enough data on them to say that they were definitely more abundant outside of the pawpaw patches, but still present, and couldn't find seeds sold. So I think Spring Beauty was something that I had information on, but couldn't find a reliable seed selling source.
the reason why you said preference?
Mm-hmm. Yeah. Yep.
If it's leave out,
Yeah, so that would be definitely a next project. I'm really hoping to get like a solid early spring data set this upcoming year, since all my plots are set up. I know what I'm doing. I'm poised and ready. Yeah.
It was the frequent caterpillars in the resistance on the all pawpaws.
So the caterpillars, I've seen maybe one, but the butterfly is that I'm not much of a bug person. Is that like a common like phenomenon that you're like? Okay, well, yeah, I can second that. The butterflies were everywhere this year. I mean, there were times I could turn around and see at least 5 to maybe 7 just sitting in one place. But yeah, no, the caterpillars I've maybe only ever seen one. So I'll make sure I keep an eye out this upcoming spring though. Ooh, gross. I have an irrational fear of moths. So I won't be reporting back on those. Oh, that's nice. All right, yeah. Yeah. So let's see here. Yeah, I do love this like canopy photo. So this big photo in the background is a pawpaw patch, and you can see it's pretty dense leaves. Let me go back to the photo I am looking for. There we go. So these canopy photos that we took with a fisheye lens, We're gonna run them through a program called Gap Analyzer that's gonna give us estimated like light measurements. So those will be considered in the analyses. Yep. I'm also hoping to go through and get some densiometer readings as well. In terms of actual, oh, you know, those light bars that get the actual level of radiation from light. I'm not going to be doing those because they're expensive and difficult. But this has been shown to be a reliable method for getting that sort of canopy cover issue covered. Okay. Are there any questions from online or anything? Okay. Well, thank you so much.
Oops, I never got. Oh, wait. Maybe. Yep, there it is. No. Probably gonna have to get it from your. Yeah.
Okay.
Sorry.
Or just plug my computer.
Flash drive, because we're. I can hand you this one if you. Yeah, let me close these first, just in case.
You can go in the meantime while I get the.
Oops, I just closed them.
You're good.
Let me. Sorry. You mind if I copy these on my desktop? Okay. Come on. It only moved to one. Sorry, everyone. Thought I dragged them both. Guess it was on there. All right. It must be under this. Okay. Yeah. They're seeing this view,
Oh, I see.
so we have to go to. Is that better, Lisa?
Meeting controls out of there. Cool. There we go. All right, so my name is Becky. I'm a third year PhD student at the University of Missouri St. Louis. And